So I want to begin my talk by sharing with you two kind of perspectives about the functional potential of nucleic acids. So this first perspective is an old one, and in this view, DNA and RNA mostly store and transfer genetic information. The second perspective is a more modern one, and in this view, DNA and RNA can do many of the same things that proteins can. So our group is interested in the second more modern view of nucleic acids, and we're trying to learn more about the interesting and useful things that DNA and RNA can do. We are particularly interested in the catalytic functions of nucleic acids, and especially the catalytic functions of DNA molecules. So we recently identified a deoxyribozyme that catalyzes a chemiluminescent reaction. So it uses a substrate called CDP star. When you remove the phosphate group from this substrate, it decomposes in such a way that light is generated. So this is a kinase deoxyribozyme. We have also kind of extended this approach to identify deoxyribozymes that generate other types of things. For example, we made a deoxyribozyme that generates a fluorescent product, and another deoxyribozyme that generates a yellow product that you can see by eye. We are also interested in developing sensors that only generate signals in the presence of a ligand. So here's one kind of simple example. This is a variant of supernova that only produces light in the presence of an oligonucleotide with a specific sequence. So I think an important question here is how might one go about making a deoxyribozyme like this? So the sort of conventional method to do this is artificial evolution. And one of the inventors of this technique, Larry Gold, is here in the audience, and this is for me incredibly exciting. So I hope you enjoy my talk. So the sort of conventional approach to artificial evolution is to first make a large library. This is usually a random sequence library. The typical size is around 10 to the 15th molecule. So this is an enormous number, a number that we don't really encounter in everyday life. This library is then enriched for some phenotype of interest, such as the ability to bind a ligand or catalyze a reaction. The surviving library members are then amplified by RT-PCR for RNA or PCR for DNA. So by doing this again and again and again, it's possible to purify very rare sequences with some interesting phenotype from these large random sequence pools. So this technology was developed about 30 years ago. It's been used many times, and it's clear that it's very general and it's also very powerful. But it's time consuming and it's difficult to learn and difficult to perform. So this has kind of prompted us to start thinking about a slightly different version of artificial evolution. So in this case, we start with libraries of 10 to the 7th to 10 to the 9th sequences. This is limited by coverage from high throughput sequencing. We perform a single random selection, and then we characterize the molecule using high throughput sequence. So by analyzing these data sets, it's sometimes possible to find the molecules responsible for some interesting phenotype. So I just want to emphasize here that an advantage of doing an experiment this way is that it's very quick, but a significant disadvantage is that these libraries are much, much smaller than those used for a conventional selection experiment. So for us, this is one of the tricks, thinking about uses where this restricted library size can still be useful. And because we're kind of worried about the functional potential of these libraries, we've also been exploring new ways to make them. So in the standard form of artificial evolution, the library is a random sequence. So this kind of library contains many, many, many different secondary structures, but a very limited sampling of each one. And in some cases, you can even imagine a secondary structure represented by a single sequence gradient in the library. One of the sort of structured libraries we've been using for single step collections is something we call a bulk library. So in this case, all molecules in the library are designed to make a single secondary structure, and a bulk in the structure is randomized. So in this particular example, N12, we can make a library in which every possible sequence is represented. A second sort of structured library that we've been exploring is called a secondary structure library. So in this case, the starting point is a motif for which the secondary structure and sequence requirements are already known. What we want to do is make a library enriched for this particular secondary structure. So in my presentation today, I will tell you about two sorts of questions that we've been trying to address using this combination of single-step selections and structure libraries. The first question is whether we can use this approach to discover new motifs. So to introduce this part of the talk, I want to show you the secondary structure of an aptamer from the literature. So this is not something we made. This is from the Shostak group. This was isolated about 30 years ago. It binds A and P. So as you can see, the secondary structure contains some stems, and it also contains a bulge. And there are many structures of aptamers in particular that have this general sort of structure. One other thing I want you to note is that this bulge is rather small. So in this particular case, 11 nucleotides on one side, 1 on the other. So what I want to show you now is the invariant nucleotides in this motif. And as you can see, they're all in the bulge. And again, this is not always true, but many times it is, that the conserved nucleotides are in the bulges, whereas the stems can change as long as the changes are consistent with base pairing. And in this particular case, the high resolution structure of the aptamer has also been determined. And as you look at the positions that make direct contacts with the ligand, again, they're in the bulge. So this sort of architecture kind of made us think, what would happen if we randomized the bulge? So we made a library that looks like this. So all library members have the potential to make these stems. We inserted this random sequence bulge of 12 nucleotides. This corresponds to 10 to the 7th possible sequences, which was kind of the target size for our library. And finally, every molecule contained a single RNA linkage shown in green. The reason for this is we wanted to select for deoxyribozymes that cleave RNA. So here is the workflow that we used. So we incubated this library under conditions to which cleavage does not normally occur. We then purified the cleaved molecules by running a single pH gel. We cut them from the gel and characterized them by hydrogen sequencing. On the right, I'm showing you the distribution of repeat numbers in both the starting library, shown in yellow, and the evolved library, shown in blue. So you can see that many sequences were enriched in this single round of selection. More importantly, some of these enriched sequences were also catalytically active. So this time course in the inset shows the RNA cleavage reaction catalyzed by one of these deoxyribozymes. So this showed that under certain conditions of this, you can identify simple catalytic nucleic acids in a single selection. So because this protocol is quite simple compared to conventional selections, it also makes it possible to address questions that perhaps aren't so easy to answer at once. So one important question when you're performing an artificial evolution experiment is what metal ions should be in the buffer. So these metal ions are needed to shield the negatively charged phosphates during folding, but they can also play catalytic roles. So people know that this is an important question that usually can't really look at this systematically. When we educated guests, in this case, we were kind of able to look at this question experimentally. So we performed a 2 single-step selections for RNA in deoxyribozyme. The first one was performed in the presence of lead, and the second was performed in the absence of lead. And as you can see, both of these selections yielded enriched sequences. We confirmed that some of these enriched sequences were catalytically active, but a kind of interesting observation is the distributions are different. We estimate that RNA in deoxyribozymes are about 1,000 times common in the presence of lead than the absence of lead. So this is perhaps not surprising. It's known that lead can promote the non-enzymatic cleavage of RNA, but this is kind of experimental evidence showing the importance of different cofactors in this particular reaction. So another perhaps less obvious advantage of this approach is that because every possible sequence was present in our library, we were able to characterize the sequence requirements of these motifs without performing additional experiments. So this slide shows how we did it for one of our RNA cleaving deoxyribosomes. We called this one Dvanotska after the Czech word for 12, and I apologize to the Czech speaker in the audience for my terrible pronunciation. So this first panel at the top shows the conservation at each of the 12 positions derived from the random sequence region. So you can see some positions like position 7 are invariant, others like position 2 tolerate some variation. We were also able to find covariations in this data set. So covariations are pairs of positions that change in a correlated way, and they're a powerful way to identify base pairs in DNA and RNA structures. So we were able to, for example, identify a covariation between positions 1 and 7 by looking at read numbers. We were also able to confirm this by making and testing for mutants. So based on this and other covariations in the data set, we were able to make a secondary structure model for supernova. So these positions here are the ones derived from the N12 region, and the rest of this is the DAFF. So to just sort of summarize the main points from this first part of the talk, we showed that function is sometimes possible in even small libraries. We also showed that we can isolate functional molecules in some cases in a single round of selection. And finally, this experiment kind of highlights the importance of cofactors, DNA active sites. So in addition to using this approach to try to discover new motifs, we're also interested in using it to try to optimize existing motifs. So to introduce this topic, I want to show you once again the secondary structure of this chemiluminescent deoxyribosome supernova. So we've spent a lot of time characterizing the sequence requirements of supernova. And based on this analysis, we think that there are maybe something like 1 million sequences that are consistent with these sequence requirements. And what we're particularly interested in is figuring out how to identify the very best one with respect to, for instance, catalytic efficiency. So the standard way to go about this would be to make a library by random mutagenesis. So you take the best deoxyribosome you have, you randomly mutagenize it at a low level, and you perform another selection. So this is a great approach when you don't know anything about a motif, but in this case, we know quite a bit about supernova. So we decided to make a library using a different approach. So we call this secondary structure library. So this is an example of a secondary structure library we designed based on supernova. So these positions shown in blue correspond to those that were highly conserved in our data set of about 135,000 variants of supernova. So we kept these constant. The positions shown in purple are ones that exhibited some variation. So for example, this position here was sometimes A and sometimes G. So we encoded this with R, which is 50% A, 50% G. You can do this using a DNA synthesizer quite easily. The base triples were encoded in such a way that each of the 3 canonical possibilities can occur in each of these positions, and every possible sequence of the triple helix was represented in our library. On the right, I'm showing you the result of a calculation we did to compare these 2 methods of library construction. So what I'm showing on the y-axis is the number of unique sequences expected to occur in the library consistent with the fold of supernova. So we first did this calculation for a series of rates of random mutagenesis. And you can see that the rate of random mutagenesis matters, and the best result is obtained, at least according to this calculation, at between 5 and 15%. However, the secondary structure library looks quite different. Based on our calculation, we think that this library contains more than 100 times more unique sequences with the fold of supernova than any of these libraries generated by random mutagenesis. So that's exciting, but this is just a calculation, and what we really want to do is characterize this library experimentally. So we made this library, and we're in the process of performing a series of single-step selections to basically identify variants with different phenotypes. For instance, we're interested in identifying variants with improved Kcat values, improved Km values, increased thermostability. So what do we know so far? Well, we have isolated a deoxyribozyme from this library with a catalytic efficiency 10 times higher than that of super. So that's cool, that's encouraging. We have also looked at the dynamics of this artificial evolution process. So what we did is take the secondary structure library and perform either 1, 2, 3, or 4 rounds of selection. We then characterize the molecules by high throughput sequence. So as you can see, as more rounds of selection are performed, the sort of separation between the starting library shown in the back and the evolved library becomes larger and larger. So that seems kind of intuitive. What we were particularly interested in knowing is where does the best sequence occur in each of these rands? And here is the answer. So after a single round of selection, the rank of this sequence is 25. And this is in a library of about 10 to the 7th sequences. As we continue to perform rounds of selection, the rank increases incrementally. We also wondered, well, what about supernova? So the best variant we had before we did this experiment. And so here's the answer. So in this case, it seems that the rank of supernova sort of levels off at something like 100. So what this suggests is that the library contains about 100 variants better than that, better than supernova. So what I just showed you was results from a selection for a single problem. But we've also obtained data sets in which we take a library and challenge it with multiple selective pressures. And here is one example. So in this experiment, we made a secondary structure library, and we selected for variants with either improved Kcat value or improved Km value. And this graph is showing the results. So each point is a different sequence. And on the x-axis, you see the read number in the Kcat selection. On the y-axis, the read number in the Km selection. So this looks sort of like a line, and I would expect that mutations, for example, that simply stabilize the structure would affect Kcat and Km similarly. But when we looked at this, we thought maybe there are lines with 2 different slopes. And when we analyzed this by bioinformatics, we found a single mutation that seems to be responsible for this difference. So we made a pair of deoxyribozymes that were identical except for this mutation and characterized them. And what we found is that this mutation seems to affect Km, but not Kcat. So if you look at this titration, at low substrate concentrations, there's about 100-fold difference in catalytic efficiency, but at higher concentrations, these curves kind of level off at about the same place. So this suggested that maybe this mutation was in the substrate binding pocket. And we did an independent experiment to kind of investigate this. So we challenged these 2 deoxyribozymes with 2 substrates and found that in the first case, the deoxyribozyme used both substrates, but in the second case, it only used one of them. So this is an example of the sort of information we can obtain from this kind of experiment. So we are interested in using this approach to simply improve our deoxyribozymes and also maybe to learn about mutations that affect different properties of the gene. But another kind of longer term goal is to use these huge deoxyribozyme data sets to make models that allow us to rationalize and maybe even predict the effects of mutations. So at the beginning, we simply want to be able to separate active and inactive sequences. But a more sophisticated approach would be to start to predict read numbers and maybe even start to make predictions about mutations that improve the catalytic properties of our deoxyribozymes. So initially, I thought we would make these models ourselves. But after talking to one of my colleagues at the IOCB, Tomasz Puszpal, he said, no, you need to use machine learning. So This is not my area of expertise, but I have a computer science student in the group, and he's kind of taking charge of this analysis. And he's also working with the people in the Plushball group. So this next slide is preliminary data, but it's also for me data that's quite encouraging. So this is a very nice collaboration between two excellent students in my group, Martin Volek and Yarda Kerfoot. So Martin made a secondary structure library based on an unpublished fluorescent deoxyribosome. He then performed a single step selection, and that result of this was a bunch of read numbers, a bunch of sequences. And what Martin was also able to show is that read number is correlated with catalytic activity. So this is kind of a breakdown of the activities of about 10 to the 7th billion for the secondary structure. Yarda has now been using different machine learning approaches to try to make a model, and the goal is to input the sequence and to get the correct read number as the output. This graph in the bottom corner shows the result of one of his models. So on the x-axis, we're showing the experimentally observed read number, and on the y-axis, the read number predicted by his model. And just to give you a little bit of information about this, he took the top 1 million sequences in the data set. He used 750,000 of them to train the model. And then he asked the model to make predictions about the remaining 250,000 that it had never seen before. So this is not perfect. I can see, for example, here's a case of an extreme outlier, but by and large, to me at least, this graph looks encouraging. Okay, so to summarize the second part of the talk, we've developed a method to make libraries enriched for a secondary structure of interest. We're using this to try to optimize our deoxyribosomes, to try to identify mutations that affect different properties of our deoxyribosomes. And finally, we wonder if these kind of data sets could be suitable for machine learning. Okay, so finally, I want to thank a number of people. First and foremost, my group for their hard work. So most of the work that I talked about today was done by Katka, Teresa, Martin Volek, and Yarda. I also want to thank my collaborators, and particularly Václav Veverka for his help with deoxyribosome structural biology. So I didn't talk about this, but we're trying to get these structures. I want to thank the director of our institute, Jan Konvalinka, for his encouragement and support. I want to thank granting agencies for giving us money. I want to acknowledge the beautiful city of Prague for providing me with a lot of pleasure and inspiration. And finally, I would like to thank the organizers for giving me a chance to talk about our research. Yeah, that's it. Thank you.